Hello everyone. It's my great honor to introduce this article to you. SynCom-mediated herbicide degradation activates microbial carbon metabolism in soils. Highlights: we provided an herbicide degradation and ecosystem regulation model of SynCom in soil. We demonstrated the effectiveness of SynCom to remediate up to 8 herbicides, to increase soil biodiversity and carbon metabolism, and to enhance soil organic matter contents. This study provides novel insights into bioremediation of herbicide-contaminated soils and thus offers a practical solution to sustainable food production. Figure 1. We screen potential keystone species for herbicide degradation by random forest model and co-occurrence networks. We identified 29 keystone species in the co-occurrence networks and compared these against the results of the random forest model, which identified 16 taxa that were most enriched under elevated herbicide concentrations. Figure 2. Morphological characteristics and functional annotations at the genomic level of strains S1, S2, S3, and S4. SynCom could efficiently promote the degradation of the co-herbicides in the culture system, and the degradation level reached more than 60%. Figure 3. Effects of SynCom on herbicides degradation, enzyme activities regulation, and nutrients promotion in soils. The SynCom was able to degrade 8 herbicides simultaneously in soil after 35 days, and the degradation processes followed first-order reaction kinetics. Sulfonylurea herbicide showed the greatest degradation, with efficiencies of 90.2% and 88.4% for nicosulfuron and pyrazosulfuron, respectively. Figure 4. Effective SynCom on soil metabolic functions at the KEGG pathways and enzyme levels. KEGG database annotations demonstrated that SynCom treatment increased the proportion of genes encoding methane metabolism, carbon fixation, pyruvate metabolism, and general carbon metabolism, when controls. Enzyme analyses confirmed that carbon metabolism increased with SynCom treatment. Figure 5. Effects of SynCom on herbicides degradation and microbial carbon pool function in soil. SynCom treatment significantly increased the relative proportions of all 12 genes. The expression of all 12 herbicide-degrading proteins was also detected in SynCom-treated soil. In addition, carbohydrate-active enzymes annotation suggested that the SynCom treatment resulted in a significant increase in the relative abundance of genes associated with glycoside hydrolases, glycosyltransferases, and carbohydrate esterases. By examining the changes in the content of the 4 amino sugars and microbial necromass carbon in different samples, we found that the degradation of herbicides after SynCom treatment could lead to a significant accumulation of microbial necromass carbon. Figure 6. Metabolic functions of key MAGs. The SynCom treatment group's metagenomic data were binned and assembled, resulting in 45 metagenome assembled genomes. The proportion of MAGs annotated as Pseudomonas, Arthrobacter, and Psychrobacter positively correlated with the degradation indices of 8 herbicides. KEGG annotation of 4 MAGs with more than 99% identity to the genomes of the SynCom strains S1 to S4, which revealed the different species occupied different functional niches in the soil. This suggests that the 4-member community exhibits effective metabolic division of labor in situ. Summary: in this study, 16 keystone species were screened after overlapping random forest model and co-occurrence networks. A 4-membered SynCom degraded multiple herbicides, stabilized colonization, and altered soil enzyme activity. Application of SynCom promoted an accumulation of KZ and necromass-associated carbon. SynCom increased microbial carbon metabolism such as TCA cycle, pyruvate metabolism, and glycolysis in soil. Meanwhile, SynCom members occupied different ecological niches and divided the labor.